All right, so we got ventilation, we've got what's going on in the external respiration, what's going on in the internal respiration. Now, here's the nitty-gritty. Here's the part that makes your eyeballs cross. All right, so you've known forever now that red blood cells carry oxygen from the lungs to the tissues, right? And you may or may not have known before this class that it picks up carbon dioxide and carries it back. And you have to have the red cells. The red cells are essentially boats with seats on them for oxygen. So you can either have not enough boats or boats with not enough seats. That's called anemia. Make sense? Okay. When oxygen diffuses from the alveolus, have an aneurysm. Okay. Red blood cell. All right, so when oxygen diffuses from the lungs, from the alveolus, into the capillary, and eventually into the red blood cell, 98% of that, 98.5%, is actually inside the red blood cell. Only a little bit, 1.5%, is actually dissolved out here in the plasma. So oxygen is a little bit soluble in water, but not much. Most of it's inside the red blood cell. Go with that? Okay. Now, what happens is, when this red cell, when this blood gets back down to here, that 1.5% diffuses out, and then a little bit that's inside the red cell goes from the red cell into the plasma. It's like an equilibrium. Yeah, it replaces it. Does that make sense? In fact, you never totally deplete the amount of oxygen in the red cell. When they go ahead.
Just a little bit leaves in time.
A little bit, yeah. As the blood is moving through those peripheral or systemic capillaries, it's dropping off a little bit of oxygen at a time. Exactly. You wouldn't want it to dump like the first 10 red cells. If they dumped all of their oxygen off, then all the tissues downstream would have to wait till they got past to get in the oxygen. You know, that way. It's like a Mardi Gras parade. You don't want to dump all your beads off at the first stop. It's trivial. Now, hemoglobin that has oxygen on it is called oxyhemoglobin. Okay. You know when they put the little pulse oximeter on your finger if you're in the hospital, whatever, everybody's seeing them, right? In fact, there's actually an iPhone app with the new iPhone because it's got the flash. You can actually, yeah, it's kind of cool. Y'all have seen House, y'all have seen medical shows. When do they start panicking? When the pulse ox, when your saturation, yeah. Yeah, they go, oh my God, because, so at 80% saturation, that means that 80% of your hemoglobin, or 80% of your blood contains oxygen, but that's not enough, okay? You want it up there in the high 90s, make sense? So that's what I'm saying, you never get all of the oxygen out of the blood. Now, how do you get the hemoglobin to let go? Hemoglobin is designed to get the oxygen. Oxygen diffuses from the alveolus into the plasma, into the red cell. Remember that oxygen binds to the iron atoms on hemoglobin. Those are the seats, if you will. One red cell has about how many molecules of hemoglobin, do you remember? Well, each molecule of hemoglobin can hold 4 oxygens. Okay. But each red cell's got like, 280 million molecules of hemoglobin? And then you've got a bazillion, a whole bunch of red cells, yada, yada, yada. Exactly. Okay. So hemoglobin is designed to get the oxygen. That's why when the oxygen diffuses, most of it gets sucked into the red cells because the hemoglobin's sitting there going, here. We don't want to be out here. So you have to get the hemoglobin to let go of the oxygen. As this blood moves from the pulmonary capillaries back to the heart, to the systemic capillaries, you have to get the oxygen off the hemoglobin. You gotta get the hemoglobin to let go. Makes sense? These are the 5 variables that control whether oxygen wants to stay on the hemoglobin or whether hemoglobin lets go of the oxygen. All right, now the first one is probably pretty obvious. This is what's called the oxygen dissociation curve. In other words, if you plot your partial pressure of oxygen here versus the percent saturation, your SATs of your hemoglobin, how much of the hemoglobin is saturated with oxygen. So, the higher the partial pressure of oxygen, the more of the hemoglobin is going to be saturated. Makes
So
sense?
pretty much the more you have, the more.
Yeah, the more oxygen is present, the more that the hemoglobin is going to suffer. So we say that partial pressure of oxygen is directly related to how tightly hemoglobin holds on to the oxygen. So, in the lungs, the partial pressure of oxygen is high. And so most of this oxygen gets sucked into the red cell, bound to the hemoglobin molecule. In the tissues, the partial pressure of oxygen is low. And so oxygen is released to go into the tissues. Make sense? Okay. So in order to get hemoglobin to let go of oxygen, you want the partial pressure of oxygen to be what?
To make it go,
To get hemoglobin to let go,
you want it to be low.
you want it to be low. So, to get oxygen off hemoglobin.
Is this our first?
Yep. You want low PO2. Now, pH, second thing. In order to get oxygen off of the hemoglobin, you want the pH to be low. So normal blood pH is what? 7.35 to 7.45. So that's this middle line. This middle line right here is basically the same line that's right here. Okay. So what happens is if pH drops, you can see how this curve shifts. If pH goes up, the whole curve shifts this direction. And what does that mean? Let's do it this way. Here's normal. At the normal PO2 of the peripheral capillaries, 20, right? Then we say that 2/3 of the oxygen has been released. Right? That's at normal blood pH. If pH is high, at the same level of oxygen, look at this, only about half of the oxygen has been released. As pH begins to drop, look at this, 3/4 of the oxygen has been released. So if you keep this the same and alter the pH, you affect the saturation. Make sense?
Saturation of the
In other words,
tissue or the
saturation of the hemoglobin. Saturation of the hemoglobin. Because this is hemoglobin saturation.
Okay.
The higher the hemoglobin saturation, the less oxygen is being delivered to the tissues because it's hanging on to it. How does that make sense physiologically? Think about what your cells are doing. Your cells are using oxygen and glucose to make ATP and CO2, right? Your cells are using oxygen, so if your cells are metabolically active, the PO2 is going to be higher. At the same time, the CO2 is going to be high. What the heck's that got to do with pH? Remember, my favorite thing to talk about, because I have lots of favorite things to talk about. It's the problem. Carbon dioxide plus water produces carbonic acid, which dissociates to form hydrogen ions and bicarbonate. So, the more carbon dioxide that's produced, the more of these that you get produced, and the lower pH. So in a metabolically active cell, pH begins to drop. Because the cell's using oxygen, it's producing carbon dioxide, so it's producing protons. And the more that yeah, isn't that cool? And so not only is the oxygen level low, the pH level begins to drop, and so hemoglobin releases even more oxygen to the cells.
Then you have to have some counteract, you don't want pH too low.
Correct, you don't want it to get too low. But don't think systemically, think at each little capillary bed, each little tissue. Remember we said that one of the things that could cause the precapillary sphincter to relax so that more blood flowed into that tissue was a drop in pH. So not only does a drop in pH cause more blood flow through those capillaries, more oxygen can leave the blood and go into the tissues. Now, this is called the Bohr effect. Why? Because it's boring. The reason this works is as more protons are available, those protons actually alter. They kind of make the hemoglobin bend a little bit, and it's almost like the oxygen pops off of it. That's called the Bohr effect. Now, here's what's cool again, because CO2, if you want to get oxygen off of the hemoglobin, you want the PCO2 to be what? What was the question? Yeah. PCO2 is the third barrier. So in order to get oxygen off of the hemoglobin, you want the oxygen level to be low, the pH level to be low, and the carbon dioxide level to be high. Well, how convenient. Because the more carbon dioxide you have, the lower your pH. And so all of this stuff happens naturally. I mean, this is cool. This is the way God made it. Or, you know, if it didn't work this way, people died and have babies. You're screwed. Or you're not screwed. Okay. Sorry. No more spring break for you. I know. Okay. So all of this stuff, the more metabolically active a cell is, the more metabolically active the tissue is, its own little microchemical environment is set up to make this whole process more efficient. The oxygen, it's like the squeaky wheel gets the oil, the working cells get the oxygen. Because just by their metabolic activities, they decrease levels of oxygen, they increase levels of carbon dioxide, which decreases pH. And as long as this happens within normal parameters, hemostasis, so everybody's happy. If it gets too crazy, that's when things get messed up. And remember that the fluid inside the cell is in equilibrium with the interstitial fluid, which is in equilibrium with the plasma. So any changes in chemical concentrations in here, changes in here, changes in here, it's all diffusion. That's why to figure out what's going on with your body, we draw your blood. Because the chemicals in the blood will reflect what's going on. Okay. Now, the second thing to remember is the enzyme carbonic anhydrase. That enzyme is present inside your red cells as well as inside your kidneys. That later. Carbonic anhydrase speeds up this process. That's a really big red cell. So this carbon dioxide is in the red cell. Process is speeded up because the red cell has this enzyme carbonic anhydrase. Cool. Okay. All right, temperature. Now, you know you've heard the stories where the person falls through the frozen lake and they're under the water for 30 minutes and they bring them out and they're able to revive them. There's no permanent brain damage, yada, yada, yada. The reason is the lower the temperature, more of the hemoglobin, more of the oxygen stays on the hemoglobin. The cells are more metabolic or less metabolically active. They're slowing down. They don't need the oxygen because they're like, it's like on those cold winter mornings where we have 4 of us. You're like I'm in my little warm spot in the bed. You stick your toe out, oh no, it's cold. When you run a fever, when you're sick, okay, your body temperature goes up a little bit. And so your immune cells need that oxygen. They're trying to fight the infection. And so again, it's the body's natural way of making it easier to get the oxygen to the tissues. So a little fever is a good thing. Again, this is just showing you different, again, given that same basic normal PO2 down here in the systemic capillaries, the difference in how saturated or not saturated the oxygen is, the hemoglobin is with oxygen, depending on the temperature. In order to get oxygen off of the hemoglobin, you want the temperature to be what? High. All right, one last thing. 2,3-bisphosphoglycerate. Well, when I was in school, they called it 2,3-diphosphoglycerate. They changed it to 2,3-bisphosphoglycerate. Di and bis means two. There's two phosphates on the second and third carbons in this molecule of phosphoglycerate. But if I happen to slip and say 2,3-DPG, it's the same thing as 2,3-BPG. It's like Krebs cycle. I still call it Krebs cycle, and they call it two other things. CAC or DCS. Now this is the way I think about this. How does this work? You know when you donate a unit of blood? Go and donate a unit of blood. Do you know how long that unit of blood lasts?
Just a few days.
Well, more than a few days. Platelets, what they do is they normally take that big unit of blood and they put this great big centrifuge, like 6 units of blood in there at a time. They spin it down and they take the red cells off. So you have a unit of packed red cells. They take the plasma off, they spin it down a little harder, and you get platelets over here, and then you have what's called FFP, fresh frozen plasma. So you actually get 3 different products from 1 unit of blood. The FFP is good for a year, frozen. The platelets are good for 5 days. You have to put them on a platelet rotator to keep stick it together. The red cells, though, anybody have any idea how long the red cells are good for? A little bit more. Depending on the anticoagulant that they put in those bags, about 45, 47 days. So a month and a half, say, roughly.
Frozen?
No. Cool, in the fridge. In the fridge. Yeah. If you freeze there is a way to freeze red blood cells, but you have to replace all the water with glycerin. But just in the fridge, like milk on the shelf. Good for about 45 days. Now it's sterile, right? There aren't any bacteria in there. It's not like it's going to go bad like milk, or good like cheese. Why did it last any longer than that? The reason is this right here, 2,3-bisphosphoglycerate. Once those red cells have been removed from the body, they're in that bag, they're no longer being fed glucose. Now remember, red cells only perform glycolysis. They don't have mitochondria, right? But they still need a little bit of ATP. They still have sodium and potassium pumps. They still have that stuff. They've got to take care of their hemoglobin. So they have to have a little bit of ATP. Well, eventually, they use up all the glucose that was taken out with them. And so one of the things that they make from glucose is this thing called 2,3-bisphosphoglycerate. What the heck's that got to do with anything? You go to a movie. Let's say there's 5 of you going to a movie. And movie's crowded. It's the Hunger Games.
Oh, yes.
Another cultural reference. Just trying to get y'all prepared for my evaluation. Except for Gary, she didn't get one. Now, let's say 3 of your friends suddenly have to go to the bathroom before the movie starts. Now, you got a problem, because there's 2 of you sitting there, and there's 3 empty seats, and what are you going to do? Because they're gone, and this is a crowded theater. Tough fun. Oh, man, you cold. What are you going to do? Oh, excellent. So, okay, I'll put a jacket here, I'll put a purse here, I'll put, a small child here. You want to save those seats, right? That's what 2,3-BPG does. It saves the seats for oxygen. So what happens is, the more 2,3-BPG that's present, the more the hemoglobin says, okay, oxygen, go to the bathroom. So in order for hemoglobin to be able to give up the oxygen, it has to have 2,3-BPG. BPG, see? 2,3-BPG. Just like the more pieces of clothing you have, the more seats you can save for your friends. Okay? So in order to get oxygen off of the hemoglobin, you want the BPG level to be high.
But what does that have to do with the blood that you're talking about?
The unit of blood?
The unit of blood.
I'm glad you asked, great segue. What happens is that unit of blood sits there, it uses up all of its glucose, and it can no longer make BPG. And so as the BPG begins to decline, if they give that unit of blood to a person, that hemoglobin that's in those red cells will not let go of the oxygen. And so it doesn't do the person any good. So, to get the oxygen off the hemoglobin, PO2 needs to be
Low.
low, the pH needs to be,
Low.
CO2 needs to be,
High.
the temperature needs to be,
High.
and the BPG needs to be
High.
You got it. All right, carbon dioxide, a little bit easier. We don't have all these variables with carbon dioxide. Here's what you need to know about carbon dioxide. Carbon dioxide just doesn't come in and stay as carbon dioxide. Once carbon dioxide diffuses from the tissues into the bloodstream, okay, it doesn't stay CO2. A little bit of it, 7% of the carbon dioxide. Remember I said CO2 is a little bit more soluble in water than oxygen? And it is. You know, divide 7 by 1.5, and that's how many more times? 3.5, whatever. Anyways. Part of the carbon dioxide, about a fourth of the carbon dioxide, is actually bound as a hemoglobin, but not to the iron. It sticks to some of the amino acids in the globin portion. Huh? Yeah, so the carbon dioxide that comes in here, a little bit of the carbon dioxide, about 7% of it stays out here in the plasma. Another 23, 20, whatever. Hemoglobin, right? Oxygen would be stuck right here on the iron in the middle, right? Carbon dioxide sticks to the globin part. It doesn't stick to the iron, it sticks to the globin. Okay? But most of the carbon dioxide actually goes out of the tissues into the red cell, goes through this process, and is converted to bicarbonate. With me so far? And the reason this happens is because the carbonic anhydrase, that enzyme, is inside the red cells. Now, the bicarbonate doesn't stay. Once it's made, it doesn't stay in the red cell. It actually diffuses back out into the plasma. Now, bicarbonate is a negatively charged ion, right? So, when that happens, chloride ions diffuse in. Basically, these two guys swap places so that you maintain the electrical neutrality. This is called, the swapping of the bicarbonate for the chloride, this is called the chloride shift. And it does nothing more than just maintain electrical neutrality. If a negative charge leaves, you need to have a negative come in. During internal respiration, carbon dioxide is basically, most of it, 70% of it, is converted to bicarbonate. Once once this red blood cell gets back to the pulmonary capillaries, the opposite happens. The bicarbonate goes in. The bicarbonate combines with that proton to make, I need to put a red cell, to make carbonic acid.
Why did you go from HCO3 to HCO2 plus hydrogen?
What'd I do? Okay, sorry.
That's a three. You just click on mine, we lost an oxygen.
Sorry.
Oh,
I exhaled that. There we go. And I leave my smelling, I'm dropping oxygen. The process goes backwards, is what I'm trying to say, to produce the carbon dioxide in the water. Okay? Because we can't breathe out bicarbonate. So we have to take the bicarbonate and convert it back to the CO2. So that the CO2 can diffuse into the alveoli and get.
So how does it go to
partial pressure, concentration. The 7% of the CO2 that's here diffuses out, and then this reaction goes the other way. Oh, don't forget, the reason this can go is because the carbonic anhydrase. This reaction is reversible.
And it's the same enzyme that reverses itself?
Which is unusual, yes.
Is that weird?
It is. Most enzymes only go one way. Why in the heck would you go through all this process? Why just leave it as CO2? What's the what's normal blood pH? I don't know.
It's got my teeth for a year now.
I know it's important.
It's been a whole year.
Now, what is perfectly neutral pH?
7.4.
No, neutral.
Oh, 7.
7. So we're a little bit on the what?
Basic side.
Basic side. This is hard. Because of this extra bicarbonate. Oh, right. No, we want to be a little bit basic. Why do we want to be a little bit basic? Because we're always producing acids. Because we're always breaking out chlorides.
That feels neutralized acids.
If you need a little bit extra, in case you get a little crazy.